Some people are kept mobile chargers. AI technology as well as now power technology also gone too much. ఏం కాదు పెట్టుకో ఐఎమ్ వెరీ ఫ్రెండ్లీ. ఐ వోంట్ రెడ్యూస్ యువర్ మార్క్స్, మీ మార్క్స్ కట్ చేయను బికాస్ ఐఎమ్ నాట్ యువర్ ఫ్యాకల్టీ. నో పెట్టుకో బాబు. వద్దా? అయ్యో పాపం ఇక్కడ కూడా ఉంది, మరి ఆమె కూడా పెట్టుకుని ఆమె ఏం చెప్పలేదు నేను. దట్ ఈస్ స్మార్ట్ ఆన్సర్. ఎనీవే నో ప్రాబ్లం. సో ఆల్ ఆర్ రిఫ్రెష్డ్ or want to get refreshed, or feeling hungry, or angry? No. Okay, so you should do something here. I am getting bored. You people are sleeping, or preparing for gate or net or in the GPAT. They are going to get good rank. Yes. Okay, so I will not waste your too much time. So I want to show a small presentation. Okay, this is very small presentation. So whatever I have done my PhD in 2016, I was awarded the same presentation. Right now I am showing you here. Okay, so I was talking about what that called target. What is target? How to identify? What is protein? So you might have got one doubt that what is target. He was shouting target kya hai yaar? Okay, the same thing now I want to discuss how to identify target, how to identify new chemical entities, and how to achieve your objectives. Okay, so now, yeah, so this was the like design and synthesis of antiviral agents for chikungunya viral infections. So I worked on the chikungunya, as I mentioned, how chikungunya and what is there, everything in detail I will explain you. In the same way, why this presentation is very important means, whenever you are doing your dissertation in B Pharm level or M Pharm level, it is fundamental to know how you have to create your project, how to design your project. Then only you can get some deliverables. So that is how you can start, that you can easily identify from this presentation. Okay, so as I mentioned, this is a beautiful building. So that building was inaugurated by, our Prime Minister, first Prime Minister. Who is the first Prime Minister? So this building was inaugurated by Jawaharlal Nehru. And you are seeing these two trees, green trees, and those green trees still there exist. And these trees are almost 70 to 80 years old. I have seen that. So that is the mark, like Bits Pilani, how it is there. The same way, these are the mark for the BIT Mesra technology, BIT Mesra. So I did my PhD under the guidance of here Dr. Venkatesan Jayaprakash and Dr. B.N. Sinha. Okay, so now the specialty of this person is, this person has did PhD under this professor. Okay, and this person, after completion of PhD, I was the first student for this person. It means he created a man force in such a way that he identified the perfect persons who is suitable for the department, and he recruited this person in the department. And right now this professor is retired last December 31st, and he become full-time professor right now. So so these are the contents, whatever the I mentioned here. See presentation how it look like, you must remember. So your picture should talk, not content. Okay, so here contents I mentioned: introduction, review literature, objectives and plan of work, experimental, and conclusion. So here this is work, whatever I want to do work, review literature, and what is the target I mentioned, and how I used the computational approaches here I mentioned, and what is the interaction of the protein and receptor is I mentioned here, how I have prepared synthesis mentioned here, and how it has been tested, and what are the publications I did. So everything will cover in this one slide. I want to deliver at this time. Okay. So coming to the introduction, as per the slide of contents, so chikungunya, it is an insect-borne viral disease caused by chikungunya virus, belong to the alpha virus and belongs to the family Togovirus, and it is spread by Aedes species mosquito, that is aegypti and albopictus. It means this is the mosquito which causes chikungunya. Okay, this was story of 2016. I am telling this was the outdated presentation, though I wanted to show you here. This was story of 2016, where according to the WHO, WHO means not who, it is the World Health Organization. Okay, so according to been diagnosed in almost 60 countries. Okay, and recent research studies were reported that vector albopictus, it is earlier aegypti mosquito, aegypti are spreading the disease. After that, in European countries, people were identified that albopictus mosquito also causing the same disease, which is responsible for spreading of this virus into the European country. So what is the difference between African continent and European continent? That's why not only Narsapur, not only Hyderabad or not only Telangana, think beyond level. People will show the movies. Their continent and their climatic changes is entirely different. European countries are very cold. African countries are very hot. Okay. The same way virus is there and mosquitoes are there. They were adapted to the cold conditions as well as hot conditions. But though they are adapted, they are carrying the same virus in cold region as well as hot region. So that was the specialty of the mosquitoes, not virus. Okay. So this was the graph how chikungunya was spread to the different countries when I started my PhD research. So I have taken this graph from the WHO in 2016. Okay. So coming to the major factors, transmission I mentioned here. So what are the major factors across the continents from the South Africa to Asia to Europe and America? So this virus, where it has been discovered, that is a question mark. Okay. After that, where it has been spreaded, that is second question mark. How it is spreading. Okay. And what are the factors? It means here travel. So what is travel factor? Why COVID during COVID time all what that called lockdown or lockdown only? Lockdown has been means imposed to ban the travel. If you ban the travel, spreading of disease will be stopped. Why? Because COVID is responsible for spreading through air, but this disease is responsible for spreading through mosquitoes. It means one person goes to Africa and where that person is bitten by the mosquito and he carries that mosquito virus through his body to different continents. That's why travel is a major factor. Vector adaptation. Vector adaptation means here vector borne I am telling. In this disease case, vector borne means the mosquito. Mosquito is vector. It means it is a transmitter. Okay. So that transmitter adaptation means earlier people were thinking about aegypti. Aedes aegypti is responsible for virus transmission. Here, albopictus mosquito was also responsible for transmission of this virus. It means albopictus also adapted the virus in his body to transmit the virus. Similarly, climatic change I mentioned. So geographically, you can see cold countries and some different factors, whatever I mentioned here. So, coming to the history. So whenever you are selecting the topic of your own interest, so accordingly you must remember you have to dig out the history of the disease or target or task, anything. Before knowing the history of the whatever the work assigned to you, it is impossible to propose the whatever your work, what you are want to do. So that gap will be there. That gap should be filled while you are designing your framework. So here history of chikungunya means this is a viral origin. Chikungunya is a fever first identified in Tanzania. So Tanzania is situated in the Africa. Okay, so this Africa again, chickpea from Aedes aegypti and human serum samples were isolated by Ross. Ross means this is a scientist first discovered virus in 1953. This was the history of the chikungunya. First identified in the 1952. Later, it was identified by Dr. Ross in 1953 and confirmed the presence of blood in the virus like blood serum. Okay, so now it was identified in the Tanzania. It was spreaded through like mosquitoes to Asian countries. After that, it was transmitted to American continent, and also during that time in the 2016, American continent was free of chikungunya virus in 2000, early 2000s and 2010s. Later, after 2015, whatever the virus to the African continent through travel history, only through travel history. Remember. So why? Because virus affected individual was traveling from one place to another place. Their mosquitoes were transmitted that virus from that affected person to different person. That's how vector adaptation was occurred in the localized. It means the virus was localized in the total across the globe. So coming to the like, for example, it was the picture in the 2014. You can see the images. What are the dark green countries were there? Those were positively affected by the chikungunya country. But in 2015, you can see. So this graph was taken by CDC. You know what is CDC? You don't know CDC? Not only FDA, you must remember CDC also. That is Center for Disease Control, which is monitored by US. Okay, after WHO, we have to get the accurate literature from CDC regarding diseases. Okay, more popular diseases. Okay, this image was retrieved from 2000 CDC in 2014. It was retrieved in the 2015. You can see the drastic changes, how countries were affected here. And what is the different, which are the different countries, how many cases were reported. I totally calculated from the literature, and what was the outbreak where the highest number of cases were reported, like that we have mentioned in 2014. So this is how you have to predict the literature from the predict the number of cases or disease prevalence from the literature, and you have to create your own knowledge using some graphical tools or Excel tools to showcase that whatever the number of digital numbers into the physical like figures. Then only people will understand what do you want to say. If you say keep on numbering, no one will bother about your numbers. But if you put one figure, everyone will see that figure and they can understand what is what. Okay. So coming, these are the numbers. Epidemiology. So what is epidemiology? Epidemiology means what? Epidemiology is a subject that deals with the disease spreading. Okay. And what is the year? I mentioned in the 1952 to 3, it was isolated in the Africa, and this is a strain name. So keep on years changed, strain names were changed, and these are the countries. These are the figures tabulated in 2013. This was the Caribbean and Cuba and some Puerto Rico. All these virus strains were isolated, and here this is the clinical features I mentioned. Though different virus strains and different countries were reported, what is the clinical feature of the virus? You must remember. What is the clinical feature of the disease? What is the feature of the COVID? Shortness breath. Oh, that Carlton, famous Carlton is there. You know, everyone is calling that Carlton. You know, at least memorize for some time. Why they were telling, keep on telling that Carlton? Shortness of breath, lungs, or you go immediately meet your physician like that. Why? Because that was the disease symptoms. The same way here, clinical features I am talking. This is acute disease most often characterized by sudden onset of high fever, severe joint pain that may exhaust you and last for days to several months. These are the joint pains. How you can, how you can means feel the joint pain caused by the chikungunya virus. So these are the symptoms of the virus, whether positive or negative. Moreover, see, chikungunya virus and dengue virus are causing by the mosquitoes only, the same set of mosquitoes. Who knows cross infection means, you know, cross infection. The person who affected by dengue may also affected by chikungunya, but physician may not able to diagnose whether that is chikungunya or dengue. In that case, appropriate treatment will not be given to the patient and his condition will be getting worse more than. So by if you know the clinical features of the disease, then only you can symptomatically treat, otherwise treatment failure will be leads to patient death also. That's why whenever you are talking about disease or anything, you must remember all these parameters. What are the clinical special features of the each and diseases here like this. So now diagnosis. So how you will diagnose the chikungunya virus? That is very important. Here I mentioned ELISA test. What is ELISA? What is RT-PCR? Reverse transcriptase chain reaction. So these two enzymes will give you the accurate result of the virus infections. Like all COVIDs were there, RT-PCR test only, everything. The same COVID, in the same way, virus also, chikungunya also can be diagnosed by these two disease means techniques only. It is difficult to identify with the symptom. Chikungunya infection is diagnosed by various serological assays. That is ascertaining existing of immunoglobulins G, immunoglobulin M, and V bodies. Here ELISA test and RT-PCR test can be done for chikungunya virus to diagnose the proper diagnosis channel, that is we call. Okay. So coming to the treatment now. So what is the treatment I am talking about? So for example, you can say I mentioned here treatment, how to stop the chikungunya virus. Medicines to prevent chikungunya virus is question mark I kept. Why? Because the specific treatment and precise antiviral therapy for inhibition of the chikungunya viral infection is not available. Till date, it is not available. Not only vaccine is also not available. Okay. The same way, the limited supportive treatment is available to reduce the symptoms caused by the chikungunya virus. Symptomatic treatment means what? This virus, you can use painkillers to get relief from the joint pain, but you are not giving medicine to kill the virus. Why? Because people doesn't know how to kill that thing. That's why treatment is under development only till date. Okay. So non-steroidal anti-inflammatory agents and antipyretics analgesic agents are directed to treat the symptoms that include fever associated with severe joint pain. It means this is purely symptomatic treatment. Okay. So it means what I have covered: introduction, how it is affecting, what are the causes of transmission factors, what is the history, what is the transmission mode, and what is the treatment. So being a medicinal chemist or being a pharmacologist or being a pharmacist, you must remember all these things whenever you are designing your project in the dissertation level. Okay. So coming to the chikungunya virus and maintenance. So virus and maintenance I am talking two different types, that is sylvatic and urban arthropod-borne transmission cycles for chikungunya virus has been reported. Chikungunya is probably originated during the maintenance of sylvatic cycles of Aedes falciparum and Africans mosquitoes in the Central East Asia. For example, if you can see this clear image, what is sylvatic cycle I am talking here? Sylvatic cycle means animals, mosquitoes. It is a cycle between the in jungle like South Africa jungle. That virus was originally discovered in the vertebrates like chimpanzees or some gorillas or some sort of other animals, and that animals were bitten by the mosquito, and that mosquito carries to the different sort of animals, and it became endemic in that region. Endemic, it means it became localized in that African continent, and that was rural cycle. Okay, sylvatic cycle means you got my point. It was identified in the animals in the African region, and it was spreaded from one animal to another animal through mosquitoes, and that cycle we are calling as sylvatic cycle. Okay, and it become endemic, it means it became localized there only. So during the rural, that rural people will generally go for their local needs of their home to the jungle. That mosquito affected that humans in the rural region, and that's why rural people were affected by the virus. And this is how maintenance here. And these rural people once visit to the urban cycle, urbanization or urban like cities, and these rural peoples again brought that virus into the urban life, and it become epidemic. It means Earlier, it was pandemic here, endemic here. It was localized to the Africa, and people keep on coming to rural peoples for their local needs, their home needs, or some other things. And after that, these people got affected by this virus, and now they went to their relatives or some other cities for visit or travel purposes, and they carried that virus into the urban life. And it means it was endemic here, but it was created epidemic, means it's outbreak, just outbreak in the different cities. Okay, and this is how it was spreaded from the sylvatic cycle to urban cycle. It means you have to remember how a disease coming from the human life to the human life. That history, if you know, then only you can know the root cause behind the disease. Otherwise, it is very difficult to identify the like medicines. Coming to the chikungunya virus genome, I'm talking. Okay, so now, as I mentioned, chikungunya virus is belongs to single-stranded RNA positive virus. So RNA virus, okay. So here it is encoded with non-structural polyproteins and structural polyproteins. These are the two sort of proteins present in the chikungunya virus genetic material RNA. Okay, and this structural protein, it is their structural protein is again subdivided into capsid protein E3. E3 means envelope protein. E2 envelope protein 6K E1. All are envelope 1, envelope 2, envelope 3, and capsid protein. All these proteins are responsible for virus entry onto the host cell. Once virus is released from the mosquito to the human body, okay, that virus will need a host to replicate its viral machinery, and it has to enter into the cell, and that cell wall cannot be broken by the virus alone. And these proteins, whatever is there, these proteins help that virus to break that cell wall, and these genetic materials, whatever these genetic materials are there, these genetic materials will be inserted into the virus host cell. So this will help these proteins to release into the virus cell. Here it means virus is virus is, let me say, so this is genetic material, whatever it is there, that is classified into nsp1, nsp2, nsp3, rdrp. nsp1 means, you know, methyl transferase. Methyl transferase, it means it is an enzyme. That enzyme is responsible for transfer of methyl in the body. Okay, thus kind of work will be done by the nsp1 in the chikungunya virus. nsp2 protease, it means this virus once enter into the host cell, it will cleave all this protein. All this protein replication will be done by this virus, this protein here. And nsp3, it means it is also called as macro domain. It means adp ribose, you know adp ribose. ADP ribose. That ADP ribose is also present in the chikungunya virus, and this protein will accommodate that ADP ribose in the host virus cell. Okay, and RDRP, it means RNA dependent RNA polymerase. It means polymerization. Chain will be polymerized using this NSP4 protein we call. Okay, this is how this is responsible. This machinery is responsible for viral replication. This machinery is responsible for viral entry. Okay. Machinery. Okay, now virus replication, how it enters into the body cell, how it goes into the from the body cell, that you have to remember. So now attachment, once this virus released by the mosquito into the human systemic circulation, that will go onto the cell surface. It will go and it will attach to the receptor present on the cell surface. It means it is reacting that whatever the capsid protein I mentioned, capsid, envelope protein, structural proteins of the chikungunya virus, that proteins will interact with the cell surface of the host cell like human cells, and after that it will become and go to endocytosis. I think you may all know endocytosis. What is endocytosis? You know endocytosis? Don't know? Engulfing cell. The same endocytosis it is. It means the virus is engulfed by the cells, and after is a viral genome here. Release of viral genome into the host cell, and transcription here. Genome genetic material will be transcribed here, and translation, after the translation it will be combined with the some other the what is 26S means, you know, 26 ribosome, 26S we call. So it will be clubbed with the ribosomes here. After that again it will club all viral machinery proteins here in the budding stage. Again it will go out from the cell, and it will affect neighbor cells. This is how virus comes and virus goes from the cell. So once if you know this machinery, then you can how we have to proceed for designing of the new chemical entities we can design here. Okay, so now before going to now, you know what is what disease, how it is coming, how from mosquitoes it is coming, how how it is transmission, what is the diagnosis, what is the treatment, how virus replication, what are the things required for virus replication, everything we have discussed. Now we have to do the literature from the when it was discovered. It was discovered in 1952. From 1952 to 2016, people have done what they have done. If you know that literature, then only we can do something. So that's why we have to do chronological literature. You know chronology? Chronological literature means systematic literature year by year, either by ascending or descending. So I have adopted here ascending literature. It means from 1952 to till date. Okay. So that literature has been done on synthetic molecules. It means chemical molecules prepared in the laboratory, natural products isolated from the plants, and vaccines. So all this literature have been done in the using some different parts of the like scientific literature source. So synthetic molecules. This is azathioprine. This is ribavirin. These are nucleosides. You know what is the nucleoside? Bases. Don't know nucleosides? Adenine, guanine, purine, pyrimidine, cytosine. All are there. This is sort of uridine base. This is also a uridine base, pyrimidine type of compound. And this is a modified version of the pyrimidine like. This is ribavirin, most popular antibiotic. Okay, antiviral also. Okay, so these people in olden days they were identified these nucleosides might affect the virus. They might kill the virus, and there what is their value here in microgram levels they mentioned. Similarly, this was the mycophenolic acid. This is a natural product compound. Mycophenolic also investigated for virus. This is Arbidol. You know Arbidol, story of Arbidol? You don't know? By seeing structure, you must remember always. Russia, you know Russia country? China, Russia. Those country actually approved this Arbidol for treatment of COVID virus. You know, that's why this Arbidol virus was approved in the China and Russia for treating the COVID virus. But somehow it become failed, because this drug is most popularly useful for flu viruses. Common cold flu viruses will be there. To treat those kind of viruses, people were using some other triazine complexes for investigated in the chikungunya virus. All these are drugs, marketed drugs. You can see all these are marketed drugs which are available, like drug repurposing. I said morning. The same way, some drugs were repurposed to treat the chicken, but somehow it was not successful. Okay, so now this was the first literature what I have gone through while I was doing PhD. This is the compound hydrazone. This compound was discovered by the same whatever I mentioned, that picture of virologist in the previous presentation. From his group, application inhibitor in 2013 it seems. Okay, so after that these are the compounds were discovered to kill the virus, and this is the natural product. Coming to the natural product, this is the whatever the polyphenol compound we can call. Whenever you don't know the nomenclature, remember how many phenols are there, how many hydroxyl groups are there, and that's why based on the hydroxyl groups people will term it as polyphenol compound. Polyphenol groups are there here, all phenols are there. Okay, this compound were tested, and these are the some terpenoid structures type of compounds are also tested, and these are the flavons. Actually here oxygen should be there, somehow I missed oxygen here. So flavon compounds are also there, and most of the time we whatever we consume from the natural food from vegetables, from those all vegetables, these compounds will be present generally. Okay, and this is the trigocharin. This compound somehow isolated from the some sea sources. You can see some, this is the bark. Those people were identified from this bark, and they isolated their compound. These compounds were become successful to kill the virus at that time. Okay, so and also these are also the same type of different trigostem. You can see here, this is different trigocharin. These are the different trigocharins. It means country. It was in America and it was in Vietnam. plant from two different countries, same plant will give you some different plant constraints. Why? Because due to their demography, due to their geographic conditions. Okay. Some of these compounds also showed inhibition. So now this was the Harringtonian. This is also same thing. And after that, I also done vaccine therapy. What are the vaccines available? And these were the vaccines were in the clinical trials. All were clinical trials. None of the vaccine was approved to treat the virus. And later, now I decided what is my objective. I have done total literature from the 1952 to 2016, and I have presented the literature data, and from that I identified that inhibitors of viral protease of HCV, HIV in the treatment of viral hepatitis AIDS makes protease as valid and potential target for designing the antivirals. Morning, I have shown you how HIV virus protease inhibitors were discovered. It means protease become popular target for inhibition of the viral replication. The same way here, I also chosen the chikungunya virus replication. It means NSP2 protease now. Now I landed with the target. So now this forms a basis for design of virus protease inhibitors for chikungunya virus. Okay. So coming to the challenges. Now what are my challenges? You can see non-availability of the X-ray crystal structure. Morning I told X-ray crystallography. It means X-ray crystallography is fundamentally useful for prediction of the protein structure. In the case of chikungunya virus, NSP2 protease structure was not available at the time. If structure is not available, then how we have to build the structure? I will show you here. Okay, not 10 minutes only. Okay, don't sleep. Sleeping, getting bored? No. If you are getting bored, let me know. No? అరే సార్ బోర్ కొడుతుందని చెప్పండి. No? Your friends were used to do, your seniors. They were always very happy. Okay, no problem. Okay. So now when structure is not available, what we have to do? Now experimental method is available to prove that anti-chikungunya agent. It means chikungunya virus protease is there. We are talking protease, but biologically it was not isolated and it was not confirmed that it will kill that same protease only. So at that time it was not available. And also this is a 3-TRK. Every protein, if you go to the literature, you type in Google PDB, it means protein data bank. Remember always PDB, it means protein data bank. In that protein data bank, whatever the target you want to identify, we can get the protein structure direct. Okay. So in this case 3-TRK. Every protein structure, remember every protein structure has only 4 numeric code. Only 4 numeric code only. No 5, no 6, no 2. It is unique. Every unique protein has only 4 numeric code. Okay. And here 3-TRK was the protease code that we have selected for protein discovery of new chemical entities, and we have provided the mechanism of action and enzyme based studies here. This was the objectives. Now we have selected NSP2 protease at that time to screen the totally publicly available databases. I mentioned databases morning, the total databases. Those databases we took, and we have taken the NSP2 protease from the protein data bank. We used computer to screen the total databases, only computer first. Okay. So now also when the structure is not available, that structure model also we developed. When protein structure is not available, we can build that protein structure by homology model here. This is homology model we call. Okay. It means we know the amino acid sequence. If you know the amino acid sequence, we will overlap that sequence with the neighbor one. चिकनगुनिया का neighbor कौन सा है? Venezuelan equine encephalitis virus or Japanese encephalitis virus. We will overlap this sequence with the other virus. We will get the total structure, and we will use that structure for the computer screening. Okay. Then design and synthesis. This thing we have done. Now plan. It means databases. We will take the protein. This is the target. This is the databases. We will use the computer here to screen the n number of compound, million number of compound. We will identify only 3 to 4 from 27 million compounds. I have taken 27 million compounds in single computer. It took for me to screen 27 million compounds 4 months. 10 years back, 15 years back story. It took 4 months to screen the total 27 million compounds. And we have taken only, I will show you whatever the compounds I have taken. And after that, we have adapted the synthesis, characterized, we have done the biological screening, and we modified something, and we discovered the anti-chick agents. Okay. Now experimental work, whatever I did. So homology modeling I was talking about. We have developed the homology model. You can see yellow color and green color. Is it visible? Yellow and green? No, visible. Okay. In this yellow, green ribbon here in this green, that green PDB was discovered through X-ray crystallography. It means it is experimental structure predicted by the instrument. It means it is accurate structure. Okay. But here in yellow structure you can see yellow structure. I have developed the model before it was experimentally validated. It means 1 year before I developed the model. After that experimental structure came and I overlapped. The structure similarity is 100%. You can see all residues are mapping. I was saying this is the phenyl, this is the tyrosine. What happened? Any problem? No?
Madam.
Okay. So you can see this overlapping. It was perfect here. Okay. So coming to the like. Yes. Hello. Somebody was actually online. Hello. Sorry. He said sorry, yaar. He said sorry, yaar. No. Okay. Anyway, no
Yes, sir.
problem.
Yes, sir. Sorry for disturbance, sir.
Yeah, no
Sorry
problem,
for
no problem. You have any
Thank
doubt?
you, sir. No doubt, sir.
Yeah, okay. So I'm carrying on. Yeah. Okay. So right now I was here. It means whatever I developed model and experimental model was 100% correct. But this yellow color model was developed through knowledge. And that green color model was developed through experiment. So whenever protein structure is not available, target structure is not available, you have to use your brain. Then only you can identify the target and you can assume the active pocket where it is present. How we can design the new chemical entities or drug. Okay. So now from that target, we identified cysteine 1013, histidine 1083, and tryptophan 1084 was important. These amino acid residues are very important to interact with the new chemical entity. It means drugs. Becoming drugs are inhibitors of the chikungunya virus. Okay. And after the identification of these residues, don't bother. I also don't know. But experimentally, some biological experiments will be there. Those experiments will determine these residues' importance in the computer and how it is there. So with the proof concept of the biology only, we will take the here. We have screened the total databases. We have filtered drug-like filter. You know what is drug likeness? You don't know? Don't know. Drug likeness. Lipinski rule of 5. Rule of 5. So this is a gate question. GPAT question here. Lipinski rule of 5. Adsorption distribution. What is there? It should have C log P like 3.5 around. And number of rotatable bonds, it should not have less than 15 like that. It was a Lipinski rule. So all, if you know, morning session I said 2500 drugs are there. Among the 2500 drugs, if you exclude the natural products, all drugs will follow Lipinski rule of 5. It means less than molecular weight 500. So Lipinski, the name, scientist name, who took all structures, he determined this rule and he kept rule of 5. And whatever that rule of 5, if you take the database, if you imply half of the databases will be eliminate the undesired structures from your computer screening. So only it will allow you drug-like structures to interact with the protein. Okay, you got my point? The same way we have developed here. These are the compounds we took for HTVS. It means high throughput virtual screening here. Okay. So now we have taken protein. We have taken the drug-like compounds here. We have screened the top 100 molecules. You again rescreened, we synthesized, and we did biological screening. So I mentioned from the 27 million compounds, I shortlisted these compounds only. You got my point? So these compounds are physically also available, but if you want to screen, you can buy them. If you want to buy, you have to spend money. If you don't want to buy, you can prepare them in your laboratory if you have synthetic sources and knowledge. You got my point? So what I have done, I have adapted these two structures for synthesis in the laboratory, and we have synthesized the compound. Whatever this compound is there, this was I synthesized and also expanded their analogs here. And this was the synthetic method. You know IR? What is IR? Infrared spectroscopy. So this is the difference between aldehyde and alcohol. If alcohol, alkyl chloride. So you have to identify how the hydrogen behaves in the CH stretch, CO stretch or OH stretch in the level of the IR. It means functional group modification, how the behavior of the compound changes with respect to, can also, you can change the some spectroscopy levels here, and we have to determine the melting points. Then after that we have to identify their NMR. NMR, you know what is NMR? Nuclear magnetic resonance. Okay, so we have synthesized compound, and we have identified the number of protons and number of carbons in the whatever the synthesized compounds. It means it will give you the crystal of, means X-ray structure of the compound. How many carbons are there? Matched. How many protons are there? Matched. Molecular weight will give you overall weight of the compound. Everything will be determined by the spectroscopy and chromatography. Okay, so now I have done another synthesis, another analog synthesis, the second identified objective, and also slightly modified. I have discovered this thing, but I modified this S with NH2. I designed some modified scaffold also on my knowledge based. Okay. Earlier I discovered only this one, but I wanted to modify the sulfur with nitrogen here. I also done that thing. So we have synthesized lots of compounds here. All compounds we have synthesized here. This was the compounds. So later we also discovered their NMR mass spectroscopy here, and also how it is behaving. See NH2. You know, morning I mentioned ketoenol tautomers. Remember, memorized? So this is, you can check here, keto form or tautoform, like some enol form or something. Hybridization state, something double bond shifting will give you imine. This is amine. This is imine. This is amine. Got my point? This is primary amine. This is secondary amine. Okay, this is imine amine form. This thing you can determine in the computer aided drug designs also. And also we have done modified the structure. I will show that thing. So now this is the scaffold identified from the first scaffold here. Whatever I mentioned, the circulated in the first square, and this is the scaffold of the second thing I mentioned. I clubbed both, identified two hits. I clubbed both pharmacophore features here, and I designed new compounds here again. You can see. So that's why how we have to use the knowledge how to design the compound, but its purpose should be fulfilled. Otherwise, designing is waste. That how purpose is fulfilled, I will show you experimentally. Okay. So now we also synthesize these compounds. The same thing story. Now biological screening. So whatever I mentioned, the morning photograph, the half of the biology has been done in his lab in Belgium. Half of the activity was done in the, like, Boris Pasteur's lab in the France. Okay, so we have used two different strains. One is Indian strain, one is chikungunya Indian strain, and another one is another strain we have used here, chikungunya. This is another strain. It means by demographically chikungunya virus two different strains. It means they mutation wise or amino acid wise they might be different. And I wanted to check this whatever the synthesized compound of them, and these were the activity. In the first series, whatever we have identified in the computer was given inhibition of the virus. It was fundamentally now identified that computer whatever given identified, and we have synthesized, and biology also proved that this compound is inhibiting the virus replication into the whole cell. It means whole cell assay means they will take healthy cells, they will inject virus into the healthy cells, and this they will give the compound to the cell, and they will check whether virus cell is getting killed or cured. If it is cell is cured, then this compound is inhibiting the virus. You got my point? So that way it was discovered that this compound is inhibiting the virus. In the second series also we have identified three different compounds were discovered as like they are inhibiting the virus, and also this is the graph of the virus how it is inhibiting. And in third case whatever I modified here, you can see I modified double bond here. You can check this whatever double bond here, CH double bond to NH2, and this work was screened in the Belgium. Earlier works was screen in the France, now this work screen in the Belgium. Though these compounds were inhibited somehow the virus in the different strain. Okay, so why this energy is very important, I will show. Okay, so these are the graphs, and this was the hybrid compound I mentioned. This is the pharmacophoric features of two different earlier provided compounds. They were also inhibiting the virus, but somehow less extent. Okay, so now this is the graph, and this is the binding modes, and you can see whatever I identified that compound. You can clearly see whatever I identified compound from the computer is in the computer, like virus means whatever the receptor level. You can see here it is making hydrogen bond. You can see yellow dotted lines. You are seeing yellow dotted line visible. This yellow dotted line means it is making interaction, hydrogen bond interaction with the tryptophan residue of 1084, whatever I mentioned. And also you can see here green line. You can see green interactions, pi electrons. It means this is also making pi interactions, and also NH2 group. NH2 group is very important to interact with the aspartic residue. It is negatively charged. This is positively charged. Positive negative charge interactions was established here. Okay. So now coming to the inactive compound. When I shifted this compound, this is the nitro position at 3 group, and it was nitro position at 4th group. So now you can see here interaction is missing. Can you see here hydrogen bond is lost, pi-pi interactions was lost. It means this compound doesn't show activity, that compound showed activity. It means you can identify the basic differentiation, fundamental difference. If you know the knowledge of the structure and protein and some drug design tools, you can run behind the activity things. Okay. So this was the some different tools here. Autodoc, small molecules have also done the same thing. So these are the same thing. So this was the same thing. And you can see whatever I told you hybridization. So whatever I mentioned, these were the two pharmacophores. This was the three active compounds. I clubbed them to single compound, and it was also interacting with the same sort of mechanism. You can see here. That's why I changed whatever the NH2. This is urea part. We can call it as urea. Thiourea means urea. Urea. NH2 CO NH2. NH2 CS NH2 means thiourea. That thiourea cyclic version is this one. Cyclic version. Acyclic version to cyclic version I converted, though it was also binding in the same pocket, same interactions, it was inhibiting the virus. Got my point? So this is how we have to provide the justifications while doing the experiments as well as doing the computational experiments. And also in final conclusion, see, whatever our objectives was there, developing a theoretical model for homology. It means before starting the structure, we have developed the homology model, means protein structure we have developed using computer. Later we matched with the experimental structure, it was 100% pure and done. And after that, that model was employed for identification of the chemical entities, and that chemical entities were discovered through computational approaches, and that compounds were synthesized in the laboratory and biologically tested, and those compounds will become chikungunya virus inhibitors. And this work we have published in the we filed patent somehow, but I lost this patent. I could not give what the doubt representation with respect to this patent. Why? Because I was not aware of that thing. That's why I filed, but it was not granted actually. And here these are the publications we made from this work. So, and also I present, as I mentioned, I presented this work in the Taiwan. This is IMEX, AFMC we called, 9th Asian Federation for Medicinal Chemistry International Symposium. I went to Taiwan and I presented this work in the Taiwan, and during that time I met whatever my project collaborator. I don't know what they were coming there. Unfortunately, I met them, and I had a chance to meet them, to explain them, and to get interaction with them, and also some mutual exchanges. And after that, my work has been done very fastly, and that's why I submitted my PhD. And this was the also when I was as a PhD fellow, I presented this work in IACT also, and this chapter somehow we retracted. And thank you. The same actually we planned this work to publish in the chapter, but later I converted this chapter into review article. Thank you. So this is a publication snapshot. Right now this its impact factor is 6. Yeah, these are the publications. and it was my acknowledgement at that time. Okay. So yeah, thank you very much. If you have any doubt, you can ask now. I think I used my time or extra time. No extra time? Got bored? Yeah, no. Your faculty or anything, you are telling anything to you? No? You should be active, yeah. Otherwise, no Facebook. Facebook era gone. Now, Reels, Instagrams. Later? Later what next? Now, no physical papers. Now all e-papers. Now online news. Next what? For that next what, you have to think in that mode, not dull mode. You have to be very self-encourage you. Nobody needs to tell anything to you. You have to get motivate yourself only. Then only you will get at least good seat in the NIPERs. Okay. I think previous batch students got NIPER seat. I think somehow I know them actually. Okay. Yeah. 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 You can ask. How vertebrates got affected by the chikungunya virus? So that's why, yeah, good question. At that time in 19 like 50s, you can sit. You can sit. Yeah. This was the question mark actually. It was how virus was affected to the vertebrates was a question mark, and somehow probable answer was that virus crossing, you can call this crossing of different animals in the jungles with a different species got reason for affecting this virus at that time. But after affecting that virus, it was discovered by the Ross. Cause of that virus, origin of virus we call, that origin was not yet discovered, but it was discovered in the area, in the year by the scientist. But how it was uh, originated from the vertebrates is still now question mark. Not only that thing, HIV also there, dengue also there. You can imagine hundreds of viruses are there. Source is Africa, almost all. Why? Their sylvatic cycle is very high. Their animal population is very high. Their drought condition is very poor countries, poor hygiene things, and facilities are not there. Everything matters. That's why climatic conditions, demographic conditions, geographic conditions, all these factors for origination of virus into the like vertebrates. Okay, that is a question. Answer is the same only. You can ask how it is discovered means discovered okay, but how it was affected to the how affected also okay, but how it was came into the globe that doesn't know. Yeah. You can go at detection. Detection is in fraction of that is in fraction. Like 1 day or 2 days we can determine. Like, but actually earlier olden days, uh, if mosquito bites, you can sit, you can sit. If mosquito bites, to know the symptom, it will take 14 days. You got my point? 14 days span of time to see the antibodies present in the like patient. Then only if he is complaining of severe joint pains, at that time doctor will refer him to go for ELISA test. If ELISA test is done, that can be done in like 24 hours. कुछ क्वेश्चन पूछे यार boys always dull. No, he is busy with smartphone I think seems. Charging is done. No, fully charged. Yes, you have to be charged. Otherwise, it is very difficult in the life, you know. No girls will come. Okay? Chalo.
Thank you, sir. Now let's all have a lunch break and gather back here by 1:40. Thank you.